Good day. I'm Nonalon M. Aspe of the Mindanao State University at Naawan in the Philippines. And in behalf of my co-authors, I present this work entitled Vermicast Analysis with the Earthworm species Pheretima losbanosensis, Bacterial Profiles for Potential Applications in Agriculture. Vermicomposting is the process of using earthworms for degradation of materials. Vermicasts excreted by earthworms have beneficial applications primarily in agriculture. Vermicasts are characterized as having higher pH and moisture content than soil, along with greater enrichment by carbon, nitrogen, phosphorus, ammonium, polysaccharides, exchangeable cations, and humus. Vermicasts were reported to be inhabited by diverse species of bacteria. Earthworm vermicasts, therefore, offer favorable effects for improving soil properties and fertility, which in turn contribute to better plant growth and yield. In spite of the known benefits of vermicasts, information on the physiological profiles and nutrient mineralization activities of their associated bacterial communities is lacking. With more than 200 known earthworm species, the Philippine archipelago is a vital geographical resource for indigenous earthworm diversity. These are just some of the many earthworm species that have been discovered in the Philippines. However, there are no published studies and baseline data yet on their vermicast and their associated bacteria. This is the first report on the bacterial communities present in the vermicast of an indigenous earthworm in the country, supporting the benefits of their utilization and potential application of their vermicast as organic input in agriculture. Indigenous earthworm species are being cultured in Folia tropica, a vermi facility in Los Baños City, Laguna, Philippines, for their vermicasts. The earthworm species cultured are identified as Pheretima losbanosensis. The vermicasts are processed, packaged, and sold in major stores in many parts of the Philippines. In this work, the microbial community in the vermicast of Pheretima losbanosensis was profiled for their potential application in agriculture. The vermicast of the popular vermiculture commodity, the African nightcrawler Eudrilus eugeniae, was also profiled for comparison. Earthworm vermicasts were assessed for their community and physiological profiles and nutrient mineralization. The results of this work will be used as baseline data on the bacterial community profile of Philippine earthworms, promote and utilize earthworms in local vermicomposting, and derive potential applications in organic agriculture. For the materials and methods, 20 adult Pheretima losbanosensis were collected from Folia tropica in Los Baños City, Laguna, Philippines. After 12 to 15 hours, fresh vermicasts were collected from the containers where the earthworms were kept. Adult Eudrilus eugeniae, or the African nightcrawler earthworms, served as control for comparison. For DNA extraction and next-generation sequencing, taxonomy assignment and diversity analysis were carried out using the quantitative insights into microbial ecology, or QIME, from phylum to species level identification. 97% similarity was used as threshold for species level identification. Diversity analysis was carried out to determine the alpha diversity metrics, including Chow 1, Shannon, Simpson, and Goods coverage. Community level physiological profile, or CLPP, of bacteria in vermicasts was also conducted, wherein average well color development, or AWCD, richness, and Shannon-Weaver index were calculated. ANOVA and post-hoc Tukey's test were performed to compare the AWCD, richness, and Shannon's index for the two earthworm species. For bacterial nutrient mineralization screening, pure isolates were screened for nutrient mineralization, including nitrogen fixation and phosphate and potassium solubilization. ANOVA and post-hoc Tukey's test were performed to compare the colorization zone for nitrogen and SI for the phosphorus and potassium values obtained. For the results and discussion, there is a higher number of reads of microorganisms in the vermicast of Pheretima losbanosensis than in Eudrulus eugeniae. Bacterial diversity is also higher in the vermicast of Pheretima losbanosensis than in Eudrulus eugeniae. The results on microbial diversity is supported by the results generated by CHIME at the phylum and family levels, as shown in the diagram. The results on microbial diversity is also supported by the results generated by CHIME at the genus and species level, as shown in this diagram. The average color wheel development and Shannon's indices of bacterial communities of Pheretima losbanosensis were significantly higher than those of the Eudrulus eugeniae. Carbon substrate utilization patterns, or CSUPs, of the two earthworm species showed that the bacterial community in the Pheretima losbanosensis have significantly higher utilization of amines, amides, phenolic compounds, polymers, and carboxylic and acetic acids. CSUPs of the two earthworm species also showed that the bacterial communities in Pheretima losbanosensis, as shown in this diagram, showed significantly higher utilization of the same compounds. Bacteria isolated from the vermicast produced by the indigenous earthworm showed higher plate nitrogen mineralization activities, as shown here on this figure. Same also goes for phosphorus and potassium utilization, rather solubilization. This list shows the bacterial species from the vermicasts reported to be beneficial to the growth of plants like rice, grapes, peanuts, pines, blueberries, duckweeds, legumes, beans, grass, and ornamental plants. And these are also the list of bacterial species identified from the vermicasts reported to be involved in soil bioremediation and the improvement of soil substrate. Vermicomposting offers beneficial applications in agriculture, primarily in enhancing plant growth and soil substrate owing to the diverse species of bacteria and high nutrient content present in vermicasts. Bacteria capable of nitrogen fixation and phosphate and potassium solubilization make the essential micronutrients readily available for plant uptake, resulting in improved quality of plant growth. Microbial activities also influence soil restoration and augment soil structure, including increased porosity, water holding capacity, and aggregate stability. The presence of diverse bacterial communities inhabiting the vermicast of Pheretima losbanosensis possess nutrient mineralization and carbon substrate utilization activities that are significantly higher than those of the Eudrulus eugeniae, or commonly known as the African nightcrawler. The vermicasts produced by indigenous earthworms are better than those of the non-native species in terms of having more diverse bacterial communities, higher functionality diversity or metabolic potential, and higher NPK mineralization activities. This highlights the presence of beneficial bacteria in the vermicast of Pheretima losbanosensis that are involved in plant growth promotion, soil bioremediation, and soil substrate improvement, which are vital in sustainable agriculture practices. Philippine earthworms should be therefore explored and harnessed for the production and utilization of vermicast to mitigate the probable threats of invasive non-native species. Thank you very much.